have their own. Uh, excuse me. Yeah, thank you. Sorry about it. So, well, I think as we all know, that the institute, after completing 14 years, would enter to the 15th year. And that's how we decided to celebrate it for the whole month. So we started on 25th with the first lecture that was on the occasion of World Veterinary Day. And Swati came and spoke about one zoonotic pathogen. Then it was followed by the celebration, and fantastically it was celebrated with a lot of team efforts. Of course, DBT also took interest and they also came forward. So with that, I'm very happy and pleased that my colleague director has taken time out and is here. So this is a continuous effort, and in next after a day we are going to have Rini from ISC, and she's a scientist and she works on an important area. So this way we have plans, but for today we are all very excited and happy, and I also welcome you on my own behalf and on behalf of the complete NIB fraternity. So over to Prasad.
So, thank you, ma'am. On behalf of Director NIAB and the NIAB fraternity, we are honored to welcome Dr. Ullas Kolthur-Seetharam. He is an eminent scientist and academic leader. Dr. Ullas currently serves as a Director CDFD and Professor at TIFR, both Hyderabad as well as Mumbai. With research interest spanning metabolic regulation, aging, genome stability, and cellular stress responses. His interdisciplinary approaches, combining genetics, molecular biology, and physiology, has led to influential contribution in aging biology and metabolic disorders. Professor Ullas is a recipient of several prestigious awards, including Swarna Jayanti Fellowship, J.C. Bose Fellowship, INSCRM Fellowship. He is an elected fellow of all the academies in India, like INSA, NASI, IAC. Today, we look forward to hearing from Dr. Ullas as he shares his insights onto metabolic and epigenetic plasticity and memory towards precision, nutrition, and health. So without further delay, I invite Ullas to deliver his lecture. Thank you.
So at the outset, I would like to thank Dr. Taru Sharma, whom I call Taru, for this wonderful opportunity. It's a pleasure to be here. Sometimes proximity always leads to the thing that, you keep thinking that you'll come at some point in time, but never come. It's like I've been in TIFA Hyderabad for 3.5 years now. I've never made it here for a scientific engagement. Of course, I've come for many other things. So, of course, I hope that this is the beginning of many such interactions. And again, thank you for that introduction. Let me just share the screen 1 second. Yes. Okay. Not working. Okay, it doesn't matter. So, what I'd like to do is that give you an overview of what we do. Of course, we do many different things, but I'll try to kind of bin it into two large focus areas. One is epigenetics, the other one is mitochondria. And I put it as precision health and nutrition, or precision nutrition and health, because I think this is not just important for humans, but also for animals. And in fact, what we understand in animals is what actually we translate to humans in most of our science that we do, right? As was introduced, I have been at CDFT as a director since last 1 year and a couple of months. My science largely still happens in TFR Hyderabad and Bombay. I'm also going to touch upon this new unit that we started about 4 years ago, and it's taken up in very nice shape now. Now, I would like to make it more interactive, so please feel free to stop me. I know you're having many seminars in the last 1 week, and probably you'll have many more. Quite likely that your brains are going to be fatigued, hopefully not. Particularly students, please feel free to stop me anytime you want to ask questions. So one of the things that we all worry about is the phenotype, ultimately, right? Whether it's a plant or an animal or a bacteria or anything that is living. And the phenotype is always a culmination of both the genotype and the environment, right? And environment comes in all different forms and shapes and sizes, whether it could be heat, it could be diet or whatever, right? And largely we've thought about how gene environment interactions happens is through modifications that happen on the epigenome. And we thought that that's... Do you have a stick?
No, we don't have stick.
Okay, so it's useful to have 18th century tools, right? So they never fail you, right? Modern technology can go wrong. Anyway, it doesn't matter. I'll use my hand. Now, modifications happen on many other proteins, right? Acylation, phosphorylation, methylation are not prerequisites or exclusive, special functions on the epigenome, but happens on other proteins. Now, I grew up in biology thinking molecular biology and biochemistry are important. Of course, molecular biology, I thought that if you understand genes, you'll be able to solve all the problems in the world. Of course, we can solve the problems, but we can only solve probably 20% or 30% of the problems. And people thought that by sequencing the genome, we'll be able to have therapy for cancer, for neurodegeneration, everything. But unfortunately, it's not. We have sequenced the genomes for many things, but still we are far away from even understanding how biology operates. So the classical view is that genes give you molecular components, but turns out that that's not necessarily the fact. In fact, people now realize that metabolites actually the ones that control molecular functions. And I'll give you an example of that in my talk. But essentially, metabolism, which is coming in the form of diet, is one of the most predominant environmental input, and that shapes how the gene flow happens and how your epigenetic modifications also get regulated. Now, biologists are far away from even understanding how energetics and thermodynamics function. I don't know how many of you students know what thermodynamics is, right? We all learned it in 12th standard, probably. Some of us might have learned it in BSc, but then we have forgotten about it. But every protein-protein interaction is thermodynamically regulated. You have an entropy in the system, you have an enthalpy in the system. All of this is actually what is regulating whether two things can come together. Your receptor ligand, vaccine efficacies, all of that determined by thermodynamics. So, but because biologists are afraid of numbers, okay, we generally say in the interviews, students will say, "Sir, math question mat puchiye, please." I'm bad in maths. Anyway, so the point is that identifying genetic and molecular components is insufficient because it will only tell you the tools that the biology operates. It doesn't tell you how the tools come together to give you a function, right? If you give a plumber the electrical tools, he doesn't know what to do because the tools are there, but the plumber is not trained to utilize the electrical tools. Similarly, you can't give an electrician a plumber tools because tools are the same. So what I mean to say is that identifying genetic and molecular architecture is important, but it is insufficient to ask how they come together in space and time to give you biological outputs, right? So this is what becomes very relevant. Now, if you think about physiology, if you think about aging, if you think about metabolic diseases, there are several processes happening inside the cell or inside the organism where multiple organ systems are doing very different things. If you want to ask such a complex question, usually labs take one protein approach or a one pathway approach or a one cell approach or a one tissue approach. Instead, we took a very conscious decision to do it very at a systems level, where we look at everything from what happens in the nucleus to what happens in the mitochondria to what happens in the cytosol, extracellular signaling, intracellular signaling, and ultimately look at how does metabolic sensing regulate molecular architecture. And what is also unique for our lab is that we work with evolutionary diverse organisms, flies, mouse, and humans. It also gives you a perspective of what got conserved across evolution, and therefore Why is it relevant? And what was different from humans to mouse to flies, so that we'll also tell you about how lifestyles could have also impacted the evolution of physiology itself. Now, of course, I'm not going to go into the details of this. The other unique aspect, like I said, is that we work with multiple organ systems. Like I said, we're not somebody who's only working with brain or liver or muscle. The whole point is you want to enjoy research, and when you want to enjoy research, animal is doing one thing, so therefore just concentrating on one tissue is actually doing disfavor to the animal itself, right? Because multiple things are happening. When you starve, liver and adipose tissue do very opposite things actually, right? And two opposite things come together to give you a survival benefit during starvation. So we do all across the tissue types, and we also work with mitochondria and nucleus. Probably we are one of the very few labs in the world which can do deep dive epigenetics and deep dive mitochondrial work, and I'm going to give you examples of that. Right? Now, if you want to think about this, you can say like, okay, what physiological context are you going to use? Are you going to use diabetes as a model? Are you going to use obesity as a model? Are you going to use cancer as a model? All of them have some underlying contribution from metabolism. So we said, we'll not study what happens when the disease has already happened. We want to ask, how does disease even happens, right? Because when you look at what happens when the disease has happened, you don't know what caused the disease, because many years have passed by, many different stresses have come together, and it gives you a molecular output. So we said, can we go back in time and ask what contributes to the evolution of the disease itself? And one of the things that fascinated us is this oscillations or transitions in biology. Remember, nothing is static in biology, right? You go through sleep-wake cycles. You go through sedentary and active cycles. You go through light and dark cycles. You go through many cycles that happens in biology. In fact, hormones are released in a cyclical manner. So these cycles can be at hours time scales, at days time scales, or at weeks or days, months time scales, based on, for example, your menstrual cycles happen periodically. Now, the question is, if I have things happening in cyclical manner, every time that system goes through a cycle, it has to reset, right? Suppose you guys go from state A to state B, I need to remember where I came from so that I go back to that particular state. If I don't go back to that particular state, if I go from A to B prime instead of B, I've lost the memory, right? I go to A prime because I don't know where I came from. And I go to B double prime because I don't know where I came from. So it becomes, what happens is that I accumulate error at every time I'm cycling, and those small errors keep pushing me to some direction which I'm not completely aware of. And that is what is causing diseases. This is typically called as an emergent property in physics. And we know now that biological systems operate like emergent properties. In fact, we have been saying this for about 4 or 5 years now, and now again the field has suddenly said that disease itself is because of an emergent property. That means you lose memory of where you came from because you accumulate very small errors, and these errors amplify over a period of time because you've lost the state, right? So ultimately, it brings down to the two most fundamental principles of anything that operates in life. One is plasticity. What do you mean by plasticity? You need to keep cycling. But there's also memory, right? Because if you don't have memory, then you don't know where you came from. So it has to be plastic, but it also has to have memory. So how do you now say what is plasticity and what is memory? I hope you agree, you understand this concept, right? You need to be plastic because otherwise you will not be able to mount a response. But you need to have a memory because you need to know what response you have to mount. That means you need to know how much to respond, but you need to know when to respond and how to respond. So all of this becomes in the context of physiology, plasticity and memory, right? Now, yeah.
That you're talking about small errors accumulate towards kind of losing during the memory or during the time period. But every part or every periodic stuff you take, everyone has its own checks and balances. They always make sure that you land into the same place again.
Yeah, so this concept of about noise and signal becomes important, and we are applying these principles of when does biology think something is noisy and when does signal emerge, right? For example, I can create noise by simply dampening the signal. Okay, if I reduce the signal, see what is noise? I have some random oscillations. Sorry, what is noise? Some random oscillation. Signal is above the threshold of that signal. I can make a signal become noisy by simply reducing the amplitude of the signal, and it becomes noisy. I can make the signal disappear if I increase the noise. But the key thing is, in biology, we don't know where the thresholds are. Okay. If you don't know the thresholds, I don't know what the corrective mechanisms are. And that's what we have actually discovered. What are the corrective mechanisms that give you these predictable oscillatory functions? Okay. Can we discover those mechanisms that make sure that plasticity is there, but also memory is there? Okay.
But biology, how can you share that you do not know what threshold is? Because the health that we define.
Yeah, that's such a gross parameter. At a molecular level, you don't know what the threshold is. Suppose if, again, I'll give you. So if you were given 3 shots of insulin, okay, every time there is a threshold signaling that is achieved, right? What is the threshold dependent on? Right? And something I'm not going to discussing, that's a paper that we also published. There's a threshold that is dependent on how much insulin you gave the last time. So what insulin you gave a last time determines the threshold to the next stimulus, depends on the threshold that detects next time.
The adaptive threshold, how you are
Adaptive again is a English word, but biologically, how does it know what threshold it has to be sensed? Do you know that if I have 100 AKT molecules, how many should I phosphorylate? Correct? How does the cell decide that it has to be only 30 AKT molecules that has to be phosphorylated, not 45? There is a memory also, there's a plasticity also. We have not uncovered those memories and plasticities in biology yet, right? Okay. So, yeah. So let me go through the talk. So I'll tell you, you probably will appreciate what plasticity and memory is. That was it okay? You can.
Constant.
There's no constants in biology.
Yes. There's no constant.
That's what I said. Plasticity and memory. There is no constancy. We all believe that when I took a Photoshop, when I did transcriptomics of ovary or whatever, I believe that's the gene expression that is causing the disease. Actually wrong. It was contributed by something else that happened much earlier, but the memory is lost. Okay. So this is just to give you a flavor of the fact that we can work on multiple things, from metabolism to energetics to PTMs to epigenomics to endocrine signaling. And also in all of these papers, you see oscillations and plasticity being highlighted in multiple different forms. Doesn't matter whether you're working with mitochondria or nucleus or cell signaling. Let's say, even if you think about genome and the gene to be God-given sacrosanct truth that that's the only way biology is regulated. Is it, students? Not really, right?
Transcription and that replication is in the context of this plasticity.
Yeah, that's what you. Maybe I'll go through some experiments and you will realize this. Let's think about genome architecture, right? If I want to transcribe the genome, I can regulate gene expression by changing the chromatin architecture. I can regulate the genome transcription by regulating transcription itself, by regulating transcriptional microRNA dependent regulation or splicing, or I can even feedback to say how much of the gene should be transcribed through epigenetic memory. Now imagine if I'm a cell, how do I know which of these is a better way to regulate my gene expression? Right? Because there is an energetic cost to each of these modes of regulation. So the thresholding is also based on what can the cell afford to regulate at that particular given point of time. If the cell wants to achieve this mode of regulation, it will choose this. If the cell wants to achieve this mode of regulation, it will achieve this. But how does the cell know which is the better way of regulation? Right? In one hepatocyte, it might do this, another hepatocyte, it might do this. So the question is that, can you untangle the rules of engagement to get that done? So that's something that, again, we have done. I'll give you specific examples, right? Okay. Now, if you think about in a much larger context, how does this plasticity and memory even get encoded in our system? Also, this is a way of showing off, don't mind, that we can also work with flies, right? We did a very cheeky experiment, okay? We put fly larvae on different diets when they are in larval stages, right? During development. After they became adults, all of them were given the same diet, right? Ideally, you should have had that the fact that because all the adults ate only the same diet, the physiological output should be the same. But no, actually, the flies had a memory of what they ate when they were in larval stages, that determined what happened at the adult stage. That means the fitness, whether it is aging, whether it's health span, whether it is ability to walk physically or physical activity, everything dependent on memory that was built from a larval stage. Tells you that the plasticity and memory is important of what happened in larval stages, gives you fitness later in life. Okay. Now, one of the best classical ways to address this plasticity in memory is through what is called feed and fast response. Feeding and fasting, right? All organisms have to go through feed and fast cycles. I'm sure you will realize that. We all eat, we all starve, right? And even a bacteria goes through what is called a feed and fast cycle. It is very well known that bacteria also goes through feed and fast cycle. This is fantastic because it's cyclical. There has to be plasticity, but there's also memory. And I know that if liver feed and fast cycles goes wrong, there is enough clinical literature. This fitness and plasticity and memory in hepatocyte isolations. Okay, so we did that entire experiment, and this is something that we proposed long time ago, and now, of course, this has come up as the really major thrust areas. This is this week's Nature Metabolism and Cell Metabolism paper, which are talking about fed and fast cycles. But actually, if you look at all my papers that we published since 2019, we all spoke about feed and fast cycles being very important. Now, let's think about transcription, right? Let's say you're a hardcore molecular biologist. Let's say you're in a starvation phase. In a starvation phase, I'm sure animal biologists will also appreciate when you're in starvation, you do what is called the rest of the organ system with glucose for surviving starvation, right? That means you've turned on a lot of transcription of starvation genes. When I go through a refeeding, remember refeeding is a very rapid response, right? I could be starved for days, months, weeks. And when I refeed within 10 minutes, my blood glucose levels are high. Is that true? That means refeeding is a very dynamic response, very fast response. When I go from a starvation state to a refeeding state, what do I do with all of these transcripts? There are a lot of these transcripts that are present. If I allow them to translate and express, it's wrong because these proteins are not required in a refed state. Are you with me? Yeah. This should not be expressed in a refed state. So what do I do with transcripts? I have to inhibit their translation and I have to also probably degrade these transcripts. So we asked what mechanisms regulate this, and we identified, I'm going to move fast a little bit because I have a lot of things to cover. We identified what are called hepatic or liver specific fed microRNAs that oscillate between fed, starved, refed, fed, starved, refed, fed, starved, refed. They show oscillation. It's a small amplitude oscillations in microRNAs. These small amplitude oscillations in microRNAs control a whole set of downstream genes which are involved for metabolism and hepatic function. And we show that this is actually important for the entire organism by doing a very cheeky experiment, which is, this is only in the liver, right? Remember, we identified liver specific thing. So what I can do is I can make adenoviral constructs and inject them into the tailvein of mouse. Adenovirus goes only to the liver, to no other organ system, and we scavenge these microRNAs. These microRNAs are oscillating. If I put a complementary DNA sequence or RNA sequence, all of these microRNAs will get scavenged. That means the targets of these microRNAs are free now. Right? So that means I've created a decoy. I've created a dummy where I can shoot. That means the real target is free to be expressed. So what we did is we scavenged these microRNAs only in the liver. When I scavenged these microRNAs only in the liver, we saw that there was a major effect on the whole body physiology. This is what is called respiratory exchange ratio, which is gold standard for physiological understanding of how feed and fast response happens. And this response was very similar to what would happen in a pre-diabetic or a diabetic individual when you already have these microRNAs being, of course, we found that microRNAs are deregulated in a diabetic state. So what am I saying? We identified oscillatory microRNAs, which are small in amplitude, and these are the ones that are regulating threshold, are trying to push to make sure that the system goes through a reproducible fed and fast cycle. If I scavenge these microRNAs, they are giving actually a disease-like state. Now, there was an interesting twist here, right? Now, I'm sure many of you will understand basic molecular biology here, that microRNAs are also RNAs that have to be transcribed. What did I say? In a starvation state, I produce a lot of mRNAs. When I refeed, the time to respond is short, and I want somebody who will come and plug these mRNAs very fast. But these microRNAs also have to be synthesized. Now, is there time for these microRNAs to be synthesized? Because your refeeding happens within 10 minutes, right? We didn't anticipate it, but turned out that we found that there is an anticipatory increase in the microRNAs. That means these microRNAs are synthesized in starvation, but kept in a precursor form. They're not processed. Only upon refeeding, they get activated and they inhibit. So that means I've loaded the gun, but I've not shot the gun. I've kept the my arms loaded, ready to fire. That means I've transcribed a lot of microRNAs. I've not processed them. When I refeed, processing happens. I inhibit the mRNAs that are required for starvation. Is that making sense? Sure? Or is it postprandial sleeping that's happening? Sure? It's post-lunch. It's the best way to sleep. Somebody is sleeping already. Okay. So now, if you want to make sure that these microRNAs are expressed in anticipation, what did we create? We actually identified a molecular anticipation. That means these microRNAs are produced in anticipation of a refeed state. When I refeed, these microRNAs are deployed as ammunition, right? Now, we asked what regulates this microRNA synthesis itself. And we now feed and fast cycles are also coupled between sleep wake cycles or light dark cycles and refeed cycles, because you typically feed when you're awake, you don't feed when you're sleeping. Now, that means there is a circadian control, there is a metabolic control. So we wanted to ask whether these anticipatory microRNA expression is metabolically controlled or circadian controlled. So we did a lot of experiments. Again, I'm not going to go into the details, but essentially what we found is that these microRNAs are actually regulated by metabolic inputs. Remember, I said that when you have mature mirs low, precursor mirs are high. When mature mirs are high, precursors so low. That means these microRNAs are transcribed, kept as precursors. When you don't need the mature mir, they get processed when you want the mature mir to be high. This is the fed state, this is the start state, because the mouse is nocturnal. During the light cycle, they're in a starvation state. During the fed state, they become active, right? So that's what we found. So we did a lot of experiments where we perturbed light and dark cycles. Again, in the interest of time, I will quickly go through it. We inverse the circadian cycle. That means we fed the mice at the wrong time of the day during the light cycle. Usually the mice only feed during the dark cycle. When you invert, remember again, the oscillation of microRNAs got inverted. That means this was dependent on the diet rather than on the circadian time scale. So we uncovered actually the mechanisms that regulate this anticipatory microRNA expression. Again, I'll go through quickly. So essentially what we showed is that, so you have actually transcription factors, which are starvation responsive transcription factors like Krebs and HNF, that go and sit and bind to the promoters of microRNAs, transcribe them. These microRNAs are kept as precursor and primary microRNAs in a starvation state, right? But after you feed, you get insulin signaling that activates this processing, and these microRNAs go and stop the translation of starvation genes in a refed state. Okay, that's what we uncovered.
It's on the feed, glucose versus liquid.
No, so we largely did whole meal. Okay, glucose does stimulate, but it does not give the same potency. So it needs the whole meal. So, but glucose is able to do it as well, but not to the effect sizes are small.
The circadian and the metabolic rhythms are not so important. It's the feed or food, whatever.
No, it's the metabolism because of feeding. It's not circadian rhythm. Yeah.
Continue what Prasad was asking. So you were talking only about the glucose. I would like you to comment on, it's a whole meal, you said. So is that the glucose behaves differently, and when glucose is planned with protein
Yes.
and fat, it's
Hugely different, very different. The body, because typically we work on what are called metabolic sensors. There are trinity of metabolic sensors: TOR, AMPK, and SIRTWINs. TOR gets activated by both glucose and amino acids. AMPK gets activated because of starvation. Sirtuins get activated because of starvation. So typically, if you look at it, they don't behave exactly the same way when you add glucose and fat and protein. They all show very distinct features. So that means the metabolites are interpreted very differently based on whether you eat only protein, only fat, or only glucose. Yes. Sorry? Okay. So we believe it's the phosphorylation of drosha by AKT. Okay, that is what is regulating it. As always happens, the student graduates, goes for a postdoc. I have to convince another student to continue this. But the problem is we do so many things in the lab. The student says, I like this than this. So this is probably someone else will come. That's an important question. We think that it's the phosphorylation dependent control of drosha activity. Yes. Yeah, so we can do that, but peripheral circadian rhythms are completely independent of the central clocks, right? So it's very well established, not our work only. There are many people which have now published that peripheral clocks are independent of the suprachiasmatic nucleus. So in the periphery actually is the metabolic cycle that tunes the circadian rhythm. So that is very well established. So yeah, we can always do central perturbation. The point is that it's metabolically controlled. Right?
So, in that case, the whole leptin story and the satiety center story, how does that fit into what we're talking about?
Yeah, so this is a slightly, we can discuss this for hours together. Satiety there's a very different functionality. Yeah, satiety at an organismal level is very different from satiety at a cellular level. So the leptin dependent satiety, which is activating through your hypothalamic centers, is largely regulating your feeding over a temporal kinetics. But that doesn't change, no, that doesn't change. It is quite independent of what happens, whether the cells have to uptake metabolites or not. Because the cellular metabolite uptake is largely dependent on the energetic status of the individual cells.
And that is delinked with the cortical centers. That's what it's.
Yes, to some extent it's dealing. Okay, except for the feeding itself. Yeah. Okay, now I will quickly go through the second. I will not have time to talk about this because I wanted to talk about something more interesting. We also found that the chromatin context itself is regulated in a fed-fast cycle. You have what are called topologically associated domains in the chromatin. I don't know how many of you have heard of this. These are loops of chromatin that are formed, and these loops are held by complexes called cohesin complex, which CTCF is one of the most important regulators. I don't know if you know CTCF is one of the most well celebrated. It's the Shah Rukh Khan of chromatin, right? I don't know if you like Shah Rukh Khan, but whatever, any Khan that you can think about. Okay, so it's that celebrity molecule which actually holds the chromatin loops together, and there are what are called topologically associated domains, and there are called subpads, which is topologically associated domains. And these domains make sure that the genes, which have to be regulated in a synchronized manner, are together in one particular domain, or brings enhancer promoter interactions together. We found that CTCF, which we used initially as a loading control, we just wanted to have a loading control. And we found the loading control itself is oscillating. Okay, led to a new discoveries that CTCF is actually regulated by metabolic inputs, and that actually determines your entire chromatin architecture, even in a fed and fast dependent manner. I'll skip this because again, this is something that we showed long ago. That means 2 years ago. Came another paper very recently, just last year, which showed that actually fasting shapes chromatin architecture. We were one of the initial people that proposed the idea that chromatin architecture is changing in a fed and fast manner, which is oscillatory in manner. How plastic it is, we don't know. It could be very plastic or it could be frozen in time. And if it gets frozen in time, it becomes memory, right? So when does the system decide that I need to be plastic, and when does the system decide that it has to be memory? I have no idea. Okay, so this is something that came about recently. But I'll go to something which will be relevant also from a human perspective. I don't know how many of you do exercise. How many of you do exercise? Very few. Very sad. Okay. Exercise, of course, is very, very important, right? Because it creates negative energy balance, and that's very important. It's not just from an energy perspective. Muscles, when you exercise, also become slightly inflamed, and it produces what are called myokines and inflammatory cytokines. These are important actually for your neurogenesis in the brain. Almost established like a God-given truth. We know that the myokines and exercise actually stimulates neuronal neurogenesis and neurocognitive functions, something that the field has done, and me and Vidita, who's my collaborator in neurobiology, we have also found, which is interesting. But let's talk about exercise. People exercise in the morning, in the afternoon, in the night, midnight, okay, whenever they want, right? So we asked, is there a correlation between nutrient availability and utilization with circadian rhythm and exercise? Because you're not equally starved at all times of the day, right? And muscle needs a lot of energy for contraction and activity, right? So we are simply asked, how are these correlated? Is there a relationship between mitochondrial energetics and muscle capacity? Now, this is important because apparently even athletes time their performance. I didn't know this before we started doing this work. Apparently, they actually, gold or silver or bronze, there is a circadian variation that affects performance even in Olympics. So the athletes also choose when they want to perform better, because that what determines whether they can get a gold medal or not. But concurrent to our study, when we started this study when about 5 years ago, concurrent to the study that we were doing, there were a lot of papers that showed that actually metabolism is very important for muscle contraction in a circadian dependent manner. But metabolism is only one part of it. I might have all the metabolites, but who will utilize these metabolites? It's the mitochondria. If I don't, it's like the engine it doesn't function, then how can the muscle function? So there was very little literature on muscle energetics, and people had not used wild type context, and of course, sedentary versus exercise was not clear, right? Now, we simply asked that question. We said, how does mitochondria behave? We performed treadmill experiment. Mouse also can be put on a treadmill. And we use a paradigm that is most popularly accepted in the field. And clearly what we found is that there is a time of day dependent change in the muscle capacity. Very similar to what happens in humans. Late active phase, the mouse have a higher capacity. Late starvation phase or early fed state, you have a lower capacity. We found this. We also found that when there is lower capacity for muscle, you also had a major dip in ATP from the muscle, total muscle, right? That means somehow energetics was better maintained when you were at a stage when you could exercise more. The energetics went down when you had lower physical activity, right? This was the hint for us to say that we were on the right path and our hypothesis was possibly correct. So we did excruciatingly detailed experiments by isolating mitochondria and doing deep dive energetics, where we looked at ATP levels, or we looked at multiple other parameters, including from number, ETC complexes, subunit supercomplex formation, substrate oxidation, turnover, mitophagy, morphology, and metabolomics. But I'm going to give you only brief because this is already published. Essentially, remember, at ZT3, you had maximum activity. At ZT15, you had lower activity. Like the muscle activity, mitochondrial activity was also time of the day dependent. This is normal wild type mice. There is no mutation here. These are just normal healthy mice. Now, we did more deep dive mechanisms. There is something called state 3 respiration. I don't know if you've heard. The state 3 respiration actually tells you the energetic potential. That means how many phosphates can you fix per mole of oxygen you respire. Do you know where we respire? Who respires when I respire? It's the mitochondria. People think lung respires. No, lung is only a channel to give you oxygen. Respiration happens in the mitochondria, right? Now, the respiration is actually coupled to energy production because it's the ATP synthase complex that is producing ATP, right? So I can calculate how much of oxygen was consumed to utilize the phosphate that I'm giving. So that is actually what is called PO ratios. We calculated all of that. And for each of those, independent of which complex that you saw, we found that there was higher capacity to produce ATP when the muscle function was higher, lower capacity to produce ATP when the muscle function was lower. We clearly found a correlation between the two. Now, fine. Is this all true? How do you know this is exclusively driven by mitochondria? Because, many things could have gone wrong. How do you know this is only muscle? Many things could have gone wrong. So we started screening for, now we use the genetic knockout to provide a mechanistic link. We did a lot of screening and we found out one of the molecules that we had earlier worked on and we had kind of ignored for a long time, unfortunately, turned out to be one of the things that contributed to this. 34 is a mitochondrial sirtuin that is expressed exclusively in the mitochondria in most tissues. And we and others have shown that it is very important for metabolism and energetics. So we knocked out 34 and we found that the time of the day dependent change in the excess capacity, remember these were the wild type mice, were completely abolished in the knockouts, right? The knockouts behaved exactly the same way as the mice that were not able to exercise at GT15, right? Now, we then correlated and provided mechanistic proof saying that actually this loss in the exercise capacity was also because of reduced mitochondrial functions. Again, if you look at the wild type, is completely lost, right? For all the measures, whether it's ATP, state 3 respiration, or uncoupled respiration, for all of those, the mitochondrial activity was very different in the morning versus evening, and it was completely abolished when 34 was not there, providing a conclusive proof that it is actually the mitochondrial function that dictates your energetic capacity, whether you can exercise more in the morning or less in the evening, right? So now we are trying to do what actually regulates this mitochondrial capacity, which is different. We don't know why mitochondria behave differently in the morning versus in the evening. Are there, again, circadian cues, there are metabolic cues, whether hormonal cues, what contributes to this? We nonetheless have clue that this is probably predominantly dependent on the mitochondrial morphology. We have done EMs from these muscles, and we found that the crystal morphology and the entire tubular morphology is different in the morning and evening. Again, what is regulating it? I have no idea. We are just trying to get deep dive into this. Okay. Any questions on exercise? Yes. No. Right. But we are doing an experiment now where we are entraining these mice for a long period and asking whether if I now make a mouse exercise only in the morning or evening, will there be a change in the mitochondrial numbers? Yeah, this long-term adaptation is something that we are doing.
Differences in the activity of complex molecules.
Nothing. I showed you that. So all of them show reduced activity in the CT15 versus CT3. Okay. So relatively, they don't seem to have any particular effect. Okay. Yes. Yeah. Can you get me some water, please? Yeah. So, it's a culmination of what we call is, I didn't have time to go into this. There is an evolutionary history and there is a life history, right? What we propose is that diseases happen because your life history is contradicting your evolutionary history. That evolutionary history is obviously genetics, okay? But your life history is not genetics, because we know that many phenotypes are non-genetic phenotypes. Obesity is typically a non-genetic phenotype, right? And people have shown that actually genes don't matter here, right? Because it's simply excess of availability of a nutrient, which leads to diseases, which produces ROS. Your mutation happens because of ROS, and ROS happens because of metabolism, right? Even if you think it's genetic, the genetic has to have a basis which is coming from metabolism, right? So therefore, it is a combination of your evolutionary history and your life history. So we propose that disease happens because there is a discord between evolutionary history and life history. Okay, I'll quickly move on to the next thing, which is, yeah. Okay, so it's one of those ambiguous molecules. Initially, people identified it as an ADP ribosylase. Then people described it as a D-lipoylase. Then people described it as a deacetylase. We propose it as a D-palmitylase. Okay, that means basically it's a deacylase. It removes acyl moieties from any protein. It could be an acetyl, it could be an acyl, it could be whatever. It's a deacylase. We show that it actually breaks thioester linkages. Fine, no, it develops diseases over a period of time, but it's born healthy. No problem. Thank you. Okay. Now let's talk about this. I'm a hardcore fundamental biologist, right? I vouch by saying that you can't translate without having any foundational, fundamental discoveries, right? People think that translation is important. Of course, translation is important, but you can't create knowledge out of void, right? Void only creates dirt. Okay, space dirt, for example, right? So you need to have fundamental discovery science that potentially has translation. So we said not everybody needs to translate, not everybody needs to do discovery, and there is a good thing that you can balance. But having done all of this, don't you think that this has human impact? Obviously, yes. So we said, can we translate? This is something that has been burning inside me since about 10 years. By when we were hopefully doing well as a lab, and I could probably project things. So first thing that we did is we started collaborating with ISRO, and we are now one of the only two biology experiments that are going on Chandrayaan-1. Okay. Now, why is this important? Space flight is a transient stress, right? We wanted to ask, how does this transient stress build a memory for later? I'll tell you, there was a all-women crew that went recently, right? Did you see that? From Elon Musk. Space tourism is going to become reality. Definitely me and Taru will not be able to afford because we are poor scientists. Probably some of you students, if you make money, you might be able to, maybe some of my other colleagues also are not as rich as going for space tourism. Maybe some of you will go to space tourism. So the question is, if you go through a transient stress exposure to space, which has multiple stresses, circadian rhythm is perturbed, gravity is perturbed, Fetwa cycles are perturbed. You also have excess radiation. So you have multiple stresses coming together in a big bang, right? Now, does the system become plastic? That means it will erase the stresses, or does it remember? So that means you're done for your life, right? So that's the question that we ask. So we are now sending flies on Chandrayaan, on Gaganyaan-1, not Chandrayaan-1. Hopefully someday we'll also send into Chandrayaan-1. On Gaganyaan-1, where the flies will stay. Actually, we wanted to send mice, but ISRO got scared, saying that, oh, agar wo katega, wire katega, ye katega, all of that. Of course, they will be in cage, but anyway, we finally narrowed down to flies because they're relatively harmless. These flies will go for a week to space, come back, and then we'll study what happens to their entire lifespan and everything from genomics to proteomics to metabolomics to everything that what happens. So asking plasticity and memory in flies which have experienced space flight. The other thing is we built Arumda, which I'm going to talk to you a little bit more about. Now, why did we make this particular unit called Arumda? All of us know all diseases run into old age, whether it's a human or an animal. We know that developmental malnutrition predisposes to diseases later in life. I'll give you examples as I go along. There is high socioeconomic burden of malnutrition. There are several neurobiology research institutes. There are several cancer biology research institutes. There is no institute to study metabolic disease and aging in the country. Cancer, neurodegeneration, everything is linked to metabolism. Okay. And therefore, if you don't have a center for studying metabolic disease and aging, we are basically done for as a country because the disease burden is so high. Again, I'll show you that. This is not just to show my face. I was young not so long ago, but this is to show that this is happening only because me and Mahendra are working together. And this is a credit to Mahendra, who's been one of the key partners here, or rather the only partner that I have in terms of taking equal responsibility. Now, Arumda has an interesting vision statement, and I would like to emphasize on this vision statement. Arumda is missioned to empower, right? Not only are we, have we dedicated ourselves to do cutting edge science, whether it is in clinical or preclinical, but we also are missioned ourselves to empower researchers, technologists, policymakers, and community health partners to tailor interventions and supplementations to populations across age groups. Remember this. This is what I'm going to illustrate in the next couple of slides. We have a physical center here in TIFA, Hyderabad. We have a virtual network. I'll show that. We have a national facility. We do a lot of human intervention. We do a lot of basic biology. We do a lot of training, but we are also doing a lot of capacity building in the country. Now, we have a website. It's slightly old. We are upgrading the website. We have about 10 more institutions added, both within India and globally, who are now partners. Essentially, the idea is that we draw expertise from all different organizations to solve a problem. People own the problems and then solve the problem. And this is one of the first units, even as an institution, to bring together basic biologists and clinicians and public health experts on this scale for the first time. People collaborate with one clinician, two clinicians, or one clinician will collaborate with two basic biologists. Maybe one clinician will collaborate with one public health expert. Here, we are thinking about multiple institutions coming together to collaborate across clinicians, basic biologists, and public health experts on a scale. And you will see what the scale is as I go through. Now, how does my work or our lab's work, which we did in terms of plasticity and memory, even have relevance to this? When disease happens, you only know pathological manifestations of the disease. But the disease could have happened because of one or more of any of these reasons. Change in microbiome is good enough to give you this. Change in malnutrition, is good enough to give you this. Malnutrition and microbiome can interact with each other, because if you are malnourished, your microbiome will be different. Then you can have an impact on body composition, your inflammation, your physical activity. All of this together will give you a disease. In reality, you don't know which of these interact with each other. These are biological causes. But these biological causes are not happening in isolation from what is happening at a socio-economic level. You might be consuming non-native diets, burgers. You might not have good dietary quality. You might be living in a particular socio-economic status which is giving you either very hygienic environment or very poor hygienic environment. Both are equally bad. You could be transiting from a village to a city. There is new urbanization, right? That is also changing my lifestyle. So when a disease happens, I have no idea what happened. How do I treat a disease? Each of these is going to be different. Can we understand the origins of a disease? And this is where plasticity and memory becomes relevant, right? Now, moreover, even if I go deep dive into biology, I have very little understanding of how macronutrients and micronutrients interplay with each other. Okay, macronutrients are your protein, lipid, and carbohydrates. Micronutrients are all your vitamins and iron, magnesium, calcium, zinc, all of those. There is very little understanding of how they interact with each other at a physiological level. Of course, when you, as a function of aging itself, we don't understand combination deficiencies. There is a lot of animal models to say protein deficiency mein kya hota hai, iron deficiency mein kya hota hai. But in humans, actually, there is a combination deficiency. When I bring in a combination deficiency, the physiology is completely different and probably non-linear. Then I can't say protein deficiency may this happens, iron deficiency may this happens, and when I combine, it will be a summation of the two. It will not be a summation of the two. It will be something else, right? We don't understand much about intergenerational effects and life course changes, and societies show nutritional transitions every 10 years. Okay, but even forget about 10 years. Even today, there are some families which are going from lower income status to middle income status to higher income status. When that happens, your nutritional transitions happens. When that happens, whatever we understood today is not going to be relevant 10 years later, because the nutrition and the lifestyle is change of the society, right? So we need to keep doing this every 10 years. Therefore, one-time study is not good enough. And ultimately, what we need is combination of high precision clinical data, deep dive biology with epidemiology come together to address this particular problem. I'm also doing a lot of exercise. Okay. Now, there is also a discovery element here. The discovery element is we still don't understand the nutrition that you take goes into major two bins. Either you build body mass or you generate energy. It's still unclear to biologists. I'm telling you because I belong to the field. How does the system know how much of glucose that you ate should go into producing glycogen and fat, and how much of it should go into producing energy? This is called resource allocation. It's an unanswered question in biology. We know the mechanisms, or we think we know the mechanisms, but we don't know exactly how it is regulated. Resource allocation is one of those fundamental, outstanding biological questions, as fundamental as discovering a gene. Okay, we don't know this. And this is what is going to happen. Now, I'll come to this in next couple of slides. Now, if you think about this, we said we have to put together a multi-pronged team, right? So therefore, we identified several preclinical collaborators across institutions. Again, this list is now many more. We've established a metabolomics and a omics platform, which has many people. We have large number of clinicians. Again, this list is very short. We are, of course, have nutrition, and we are going to hire more people in these domains. If you know people, please ask them to come and approach us. We said, why is this plasticity in memory even relevant, right? Now, there's this concept of double burden of malnutrition. I was completely ignorant and oblivious to this fact just 4 years ago. I was working on diet, I was working on metabolism, but I had rarely heard what is called double burden of malnutrition. Double burden of malnutrition, this is a WHO definition, can be defined at multiple levels. At an individual level, you can be obese, but be micronutrient deficient. This is double burden because you're having the disease of obesity, but you're also having micronutrient deficiency, which is double burden. Double burden, the other definition is developmental malnutrition increases the risk of adult diabetes, obesity, cardiovascular disease, neurodegeneration, and cancer. Pretty much every disease that you think about that happens in adulthood, the risk increases substantially if you have developmental malnutrition. That means the person pays price twice in his or her life. When you are young and when you are old, double burden. Double burden at a household and a society level is you have individuals who are extremely lean and thin, or you have individuals who are extremely obese. These are also unproductive people. Nothing to, I'm not saying they're useless people, but there is a socio-economic cost of having malnourished people. There's also a socio-economic cost of having obese people. So that means they pay a price at an individual level, but also the society is paying a big price together, right? Now, there is also something called triple burden of malnutrition. That means, I don't know how many of you know, many non-vegetarians are B12 and vitamin D deficient. You know this? You would have expected, no, I'm eating all the nutrients, why am I not? So we understand very little about how nutrients get absorbed and utilized. Okay. Resource allocation is also something whether liver should get more vitamin D or muscle should get vitamin D. Do we know? I don't think it's known. Do we know whether VDR activity is more in the liver or in the muscle? We've knocked out those genes and found that that's important, but we don't know how their activity changes during all your life cycle. So triple burden of malnutrition is very important. Okay. Now, how does this matter for the country? This is important. I'm going to take 10 more minutes. I'm sorry, I know probably I'm keeping you from chai. Okay. This is very important because this impacts childhood growth. Malnutrition single-handedly impacts the neurocognitive functions. That means these children are not dementic. They're not, they don't have neuro diseases. Their IQ levels are low. So what are we doing as a society? If I or you were to be born malnourished, my and your IQs will be lower. If my and IQs are lower, we will always earn less. I'll show you that data. If I and you earn less, our children will continue to be in a lower economic strata because we'll never be able to afford higher salaries. Our children will never be able to afford higher salaries. So that means it is directly impacting the economy of the country. Right? And this is not my fancy idea. I'll come to this. Now, why is this particularly relevant to do this now? In fact, this should have been done 10 years ago. Whatever, didn't do it. The median age of Indian population today is around 15 years of age. Okay. Amritkal Bharat, or let's say 2050, the median age will be around 40-45 years. At 40-45 is when you have increased risk of cardiovascular disease, obesity, and diabetes. What did I tell you? That malnutrition during early life increases the risk of diseases later. That means this population, when they become 40-45, will have higher risk of developing diseases. That means my worry is that by 2050, you'll have pandemic proportions of cardiovascular events where people might start dying on the street, just dropping dead because they were malnourished. And also, there is a second hit. During their adulthood, they're also abusing because they're not exercising. Okay. And they're probably eating a lot of junk food. Right. So therefore, this is worrying. And if we don't take care of this now, our country will be much worse when we are at 2050. Now, the way I sell this to the government or convince to the government why this is important is because of all of these reasons. By 2030, around 500 million youth will be there in India. If a significant portion of this youth is malnourished, imagine the disease burden later. It's known that malnourished children earn about 20 to 30% less in adulthood. That's what I told you. The economic burden of childhood malnutrition is estimated around $30 to $40 billion by 2030. But we have not been able to even come anywhere close to understand the economic burden of these children becoming obese, diabetic, or having cardiovascular diseases because it's a lifelong issue. And therefore, it becomes relevant for us to ask this as a country, and we have to ask now. Now, people think that by becoming a rich country, by becoming an advanced country, we'll solve all malnutrition. Actually, US also has malnutrition. Malnutrition is different from undernutrition. Okay, malnutrition is either excess calories or improper calories or lack of macronutrients. And the obesity you saw, I don't know if you saw this, there was a recent Lancet paper which showed that there has been astronomical rise in the obesity incidence in the US in the last 5 years than the previous 5 years. Okay, it's a very bad number, and we can't solve this. Now, I also used to think that people who are poor are probably healthier because they're not consuming McDonald's and chips and all of that, right? Actually not. We work with rural hospitals, and it's really sad to know that rural children are not able to afford milk. Please, something, NIB, do something. Okay, they're not able to afford milk, but they can buy a fizz drink. 80% of kids don't drink enough milk in their mid-childhood, but 80% of the same kids drink at least one fizz drink in a week. Because local Coca-Cola versions cost only 5 rupees. Okay, chips is cheap to eat. That means they're extremely malnourished, right? Now, words, all our RDAs, you know what an RDA is? No, is there an RDA for? Yeah, right? Is there an RDA for cattle? There should be. There's no? Okay.
Species wise.
Exactly. Now, our RDAs, we don't take into account genetic, dietary, and geographical diversities. We are a diverse population. We eat different things. Our activities are different. I'll tell you, somebody coming from Himachal to Bombay, eating the same diet increases the risk of diabetes twice or obesity twice because you're coming from a cold temperature to a thermoneutral or a warmer temperature. If you come to a warmer temperature, your adipogenesis, or let's say bronadipogenesis, is low because there is no thermogenic potential. You don't need to produce any more heat, right? So the same nutrients will increase the risk of obesity twice because you're not burning your fat that you're supposed to burn to keep your bodies warm when you were in Himachal, right? So therefore, it actually also causes problems, and there's a lot of internal migration that's happening in the country.
Cultural and traditional practices of Indian-based diet system that is practiced, these are possibly taken care of.
Yeah, but we have lost those. No, that's the problem. It's amazingly bad to see even in rural. Well, we work with Kalyani, we work with Maharashtra Rural, we work with CMC Velar, I'll tell you. Okay, we work with tribal populations in Orissa and near Mysore. Increasingly, native diets are lost. Everybody is eating the same rice and wheat. Okay, so we've lost the memory of that. Okay, now why is this also alarming? There is high prevalence of malnutrition in India. If you look at these maps, okay, it's worse than sub-Saharan Africa. In fact, in Africa, you have what is called severe acute malnutrition, which is severe malnutrition. There is typical pot belly, and you must have seen those things. In India, we have what is called MAMS, which is moderate acute malnutrition. Moderate malnutrition is not visible. You think I'm healthy, but I might be malnourished. So that means it becomes worse. Second, everything is based on BMI, body mass index, worse, particularly for Indians. You think I'm lean, but I could be physiologically obese. What do I mean by that? My muscle mass to fat mass ratio might be skewed. In fact, that's more relevant than body mass index. You could be healthy obese as long as you have right proportions of muscle mass and fat mass. So just because somebody has low BMI doesn't mean that he or she is healthy. In fact, we have very high incidence of cardiovascular events in lean individuals. There's something called lean diabetes, which is particularly relevant in Africa and Asia, not in the West. Okay, and we don't understand what happens. So most of our clinical assessments are based on BMI. We have not had a public health level, understanding of body composition, right? Now, if you look at the numbers from government, this is NFHS data, this is the National Family Health Survey data. In some states, you have malnutrition, which is as high as 40%. Best states have malnutrition, which is close to 20%. 20% is still high. The average for national malnutrition is about 33 point something or 35%. 35% of our population is malnourished, but only based on BMI. If you include body composition, I'm worried that these numbers will be much worse because even healthy individuals might become physiologically obese, right? This is another concern. Now, how do you address this? It's a complex problem, right? Now, again, goes back to plasticity and memory of biological systems. There's a concept called DALI. I don't know how many of you have heard of DALI, which is called disability adjusted life years. I have to learn all of these in the last 4 years, right? Learn a little bit of epidemiology, learn statistics and blah, blah, blah. It's interesting because you're doing something new, right? So DALI is the number of years you lose because of early mortality or the number of years you lose because of a morbidity. That means somebody who has diabetes and has neuropathy and let's say myopathy or retinopathy will be suffering and has number of years lost. If you look at what contributes to maximum DALI, it's childhood and maternal malnutrition. That means what you ate and what your mother ate when she was pregnant is dictating whether you will pay a price later in life. Remember, it's the double burden. Now, if you combine that with dietary risks, high fasting blood glucose, high cholesterol, this number will be much higher. That means what happens in early childhood, these could be your adult problems because you're not exercising, right? Many of you are not. That's what many people didn't raise your hands, right? If you combine childhood malnutrition with lack of exercise and high cholesterol, this risk will be much higher, which imagine what's going to happen to our population and also as individuals. This is really amazingly bad. Now, by becoming a rich country, are we going to solve it? Absolutely not. This is WHO data, which shows that the amount of sugar-sweetened beverages that are consumed from 2007 to 2017, it's almost doubled. By 2024, I would imagine the graph will be somewhere here. It means as we are becoming a richer country, we are consuming more processed diets. That means this is only going to get worse and worse as we talk. Of course, advertisements are not helping. Down below is the number of advertisements for confectionaries, right? Now, what is the scope of vision and scope of Arunda? It started, during peak COVID. I just finished COVID Zero survey. I have the unique honor and distinction of doing one of the largest Zero surveys in the world. We published actually a Lancet Global Health paper, even though I'm not an epidemiologist. And during that period, we inaugurated Arunda virtually because we were in the middle of COVID. And later, of course, we had an MOU between DAE and DBT. Now, if you ask me, again, going back to basic biology, we have, let's say, growth trajectories and anthropometry, BMI, body composition. We know there are deficiencies, either real deficiencies or hidden hunger. But really, from a biology perspective, we understand very little about systemic metabolism, energy expenditure, and substrate utilization. For example, mitochondria can derive energy from burning carbohydrate, protein, and fat. Right? Do we know what it chooses to burn? And when does it choose to burn? If all of us given exactly the same number of calories of protein, carbohydrate, and fat, some of us will build fat, some of us will become obese, some of us will not become obese. That is related to also our energy expenditure. So very few people, even in the nutrition world, worry about energy expenditure, right? So we said, can we address these things? One of the main motivation is to also ask long-term implications of inappropriate intervention. For example, if you feed the kid the right diet, but it's wrong for the individual, so it means, let's say you take a malnourished kid, I'm feeding nutrition, giving midday meal skill or whatever, the kid is gaining weight. Do I know if the kid is gaining muscle mass or fat mass? If the kid gains muscle mass, it's good. If the kid is gaining more fat mass, I'm actually inducing childhood obesity, but I'm happy because the child is gaining weight. So I'll remove that child from my bad data and say that, oh, child is looking healthy. Looking healthy because of BMI is very different from being healthy because of your body composition. So that's something that we want to do. Of course, we want to do predictive outcomes. This is something that we would like to ultimately also collaborate with potentially with NIAB to ask, can we go back to native crops and agronomics, which includes animal products? Can we feed cattle D2O, make milk, which is labeled with D2O, feed it to humans and ask how does metabolism happen? That's one of the favorite things that I would like to do with NIAB if possible. So we do a comprehensive approach. I've told you this. Now, one of the things that we realized when we started this is, when I came back to India, I was probably one of the only people who worked on, I was an epigenetics guy. I became mitochondria and metabolism 5 years after I started my lab. This for the students, I was a mouse guy. I had never touched a fly. I used to hate the smell of fly food, I came back and I realized I didn't have an animal house. Then we started working on flies. Then realize that if I were to work on metabolism, I cannot ignore, if I were to work on epigenetics, I can't ignore metabolism, therefore I can't ignore mitochondria. So we learned mitochondria on our own after 6 years after we established the lab, right? That means when I was doing my science, there was no centralized facility to provide any service, right? So, of course, for Arunda, basically you need a metabolomic facility. But we said instead of doing it for ourselves, let's make it as a national analytical facility for nutrition and metabolism research. Why should it be serving only me? It should serve all the people in the country. That's something that we did. I'll tell you about this and that in the next couple of slides. Now, the National Analytical Facility is already established. We built a new building, brand new building in the last 8 months in TIFI Hyderabad. It has several labs. And this is the first state-of-the-art platform to provide services on this. The second thing is stable isotope platform. Now, I emphasize on the importance of body composition. Do you know how body composition is measured? Anybody? DEXA? Have you heard DEXA scanning? Yeah. Okay. DEXA scanning is typically used for, or something called BIA, bioimpedance measurement, right? You stand like something like a weighing machine and you hold something. There is an electrical resistance that happens. That resistivity is based on blah, 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 blah. So that gives you this thing. But BIA is extremely erroneous. DEXA is precise to some extent. DEXA is expensive, present only in tertiary or secondary healthcare centers. DEXA means X-ray exposure. You don't want a person to go through multiple DEXA scans in his or her life. I didn't know 4 years ago that heavy water, you know what heavy water is? Deuterated water. All of us, pregnant mother, newborn kid, 90-year-old individual, person lying on a ICU bed on a ventilator, all of us can be given D2O. And to the amounts that are given, it's absolutely non-toxic because it's non-radioactive. Even this has some D2O in it. Okay. That means the amount of D2O gives you the total body water. Total body water gives you an indirect measure of muscle mass and fat mass. That means I can measure body composition in Anganwadis. That's exactly what we are doing. In people who cannot come to towns or cities, and we want to make sure that affordable diagnosis is available at the doorstep, right? Now, what do I have to do? I can drink D2O, collect the saliva, which is non-invasive. I can just spit into a tube, get it back to the lab. D2O water doesn't degrade, right? Does it degrade? No. I can keep it at whatever temperature for however long. I can bring it to my lab and measure amount of total body water and give it. The most important thing is IAEA, which is the International Atomic Energy Agency, which I thought was do you know who regulates who can make nuclear bombs in the world? IAEA. Do you know there's something called Non-Proliferation Treaty? Guys, you have to know this. For students, this is current affairs. India has not signed what is called a Non-Proliferation Treaty. That means, we are in principle okay to give our atom bomb to someone else, right? It's called Non-Proliferation Treaty. So IAEA used to do this. Now, IAEA, which is a UN body, actually has a lot of program on nutrition. In fact, here you see chicken, which actually has been fed with D2O. They actually also feed cattle with D2O to make meat and milk. They grow pulses using D2O to label nutrients so that I can look at this. So if IAEA can do it, we said who's the equivalent of IAEA in the country, which is BAE. So we built what is called a Siemens platform, stable isotope methods and imaging analytics for nutrition science. Of course, this is useful for plants, humans, wildlife. In fact, we support a large number of wildlife studies using this because that's the only way to measure their physiological fitness in the wild. Because I can't capture them and I can't do experiments, right? I can make them drink D2O and then collect the samples later and do studies. Now, the other thing is, remember I said that energy expenditure. How do you measure energy expenditure? I showed you what is called respiratory exchange ratio in mice, correct? Respiratory exchange ratio is a gold standard for energy expenditure. In humans also, we can put them in a cage, which is like a fancy hotel room. In fact, it's very, very fancy. It's like probably a Trident hotel room, right? Now, these facilities are present only in rich countries. Let's say largely in Western countries. About 20 such installations are there in the world. In my knowledge, none in low and middle income countries. So we said, if we can't set up those facilities, how can we do cutting edge research for human science. So we established, or we are rather establishing, human metabolic chambers in CMC Vellore for the first time in the country. And for that, we got a CSR grant from Hindustan Unilever. I went and spoke to CMC Vellore director. I don't know if you've been to CMC Vellore. It's the top-notch, probably Johns Hopkins of India. One of the best clinical research centers. Every inch of space is worth saving 20 people's lives because, you know, they have huge patient load. The director gave 2,000 square feet of space. They said, like, Ullas, you can probably, I mean, with that 2,000 square feet, maybe I can save 20 people's lives. But with this chamber, maybe I'll be able to transform and give more preventive medicine rather than therapeutic intervention. So we are building these chambers, and that's going to be inaugurated by August or September of this thing. The orders are already gone. Next, if you want to study this, I have to capture what happens throughout life course, right? Remember, because I don't know exactly when does the disease start or when does malnutrition start. So we've done a large number of studies to look at what does a normal healthy individual look like. So we took in 3 sites in rural hospital, CMC Vellore and AIMS Kalyani. We took lean males and females, normal BMI males and females, overweight males and females, and obese individuals. That means we first classified them as BMI. We made sure that they didn't have any diseases by doing thorough clinical investigations. They were not even pre-diabetic. I'll tell you why this is important. And now in this, we are comparing how does BMI correlate with body composition, how does their inflammatory cytokines look like, and what is their systemic metabolism. But what came out as a bad surprise for us is to get 20 individuals who are normoglycemic healthy in the age group of 25 to 45, we had to screen close to 60 to 80 people. That means 2/3 of the population is already pre-diabetic in the age group of 25 to 45, which is alarming. That's what I was saying, right? This is going to hit us like a blast from somewhere when we reach some, in 10 years. So it's really sad to look at so many pre-diabetics in the population. I'll come to this. We are doing a lot of work on childhood malnutrition. We started a major initiative on gestational diabetes. There's a major project that we are looking at adolescent girl malnutrition. In fact, it's the most ignored problem in the world, especially in India. We think that we have to take care of neonatal nutrition. There has been now understanding that gestational nutrition is important, but actually this has to start when the girl is at the reproductive age. If she is not fit, you're not going to get a healthy BP at all. And worse, because of pregnancy itself, which is also a metabolic cost, that woman is going to pay a price later in life. That could be probably a 4th burden, right? Because you have a double burden, a triple burden, and a 4th burden. So we started this. In fact, for this, Gates Foundation is funding us close to $4.5 million because it's one of the unique studies anywhere in the world that we are doing, right? Now, how do we say this, right? What do you mean by double burden? Remember, I said double burden is coexistence of leanness and obesity in the same population. So we took data from what Gagandeep Kang and Bina had done in this particular cohort called MALID. And we looked at how to simply, if I go back and retroactively analyze the height and weight of children, people are born with some distribution of weights and heights. At 2 years of age, most of them become normal. But what is surprising is that by 4th and 5th and 6th years, you start seeing obesity set in and severe thinness setting. Right? That means the same children who were normal at 2 to 4 years of age actually are becoming double burden. Right? Now, if I have to intervene, this is the first study. We have sent it to a very leading journal for publication because in my opinion, there has been no study which has asked when does double burden set in. We saw that the double burden sets in as early as 5 years of age, which is scary. Imagine this is semi-urban population. This is not your rich Aparna Sarovars or villas population, which is where I think obesity will be here. This is all peri-urban population, slums, slightly middle income, lower middle income. You have a high burden, about 12% obesity and about 17 or 20% severe thinness. And these are, and I don't even know how this will, project as we go along. We have done deep dive metabolomics in this. I'm going to skip this, where we identify signature metabolites that are causing this, associated with this. Whether it is causing or not, we don't know. So we do multiple sites. We are working across the country. Of course, our representation in the north is low because we want to be very careful in identifying our clinical partners. We work with multiple things. We're asking multiple questions. Again, in the interest of time, I'm going to skip this. But essentially, I'll give you a snapshot of exactly why this is relevant. The other thing is, we went to ICMR, and Dr. Rajiv Bal had just taken over as the Director General of ICMR. Rajiv said, Ullas, you're tackling Ullas and Mahendra. You are tackling very fundamental problem of malnutrition. But I don't know who is a normal kid, right? Because, for example, Rajasthanis and Punjabis, my favorite example, I could be wrong. Punjabis are generally taller. Rajasthanis are generally shorter, right? If you look at the same age, one will look thin, the other one will look obese. But you're feeding them the same diet, right? So we don't know what is the normative reference standard for childhood growth, right? Most of the data is coming from the Caucasian population. Even India, it's largely based on a population in Delhi. It doesn't capture the genetic or dietary diversities. So with ICMR, we got, Rajiv said, this is a very important problem. Can you help me define what is normative baseline? I said, absolutely, yes. We got a Center for Advanced Research grant from ICMR. This is close to 15 crores. We started doing this study. And why is this relevant? And why is this important? Again, going back to my fundamental biology, right? There's this concept of metabolic maturation, and there's this concept of anabolic resistance. For example, when people get old, they lose muscle mass. Okay, sarcopenia is a big problem in old age. Okay, I don't know, probably animals also have sarcopenia, right? When sarcopenia happens, how do you increase muscle mass? No matter what you do, muscle doesn't seem to grow. There's anabolic resistance. Anabolism is to grow. Resistance is it doesn't grow. We don't understand why anabolic resistance is happening. Essentially, it's the concept of those two will give you either normative individual or you will have an underweight or an overweight individual because somebody will be this or that way. Now, were we proposing something that was completely artificial? No. A lot of research in the recent past has actually shown that metabolic maturation is important in dictating postnatal growth. Makes sense if you're a biologist that how will I grow if I don't have raw material to grow? I mean, can you build a building without sand and stone? No. What is sand and stone? It's your nutrients. If you don't have nutrients, you're not going to build body mass. That means you're not going to grow, right? So it's in retrospect, it's an obvious question. So now we are doing, deep dive metabolomics. We are looking at secretomics to even look at what is called somatotropic axis. We are doing macronutrient composition studies. We are doing body composition. Also, capturing a lot of epidemiological data from close to around 8,000 children across the country. This is the first time where we are going to get not only anthropometry, which is height and weight, but also a lot of biological insights, which will be very useful. So we also do a lot of training measures. And please, you're most welcome. We organize what is called Arumda Symposium. This year it's going to happen in July. In this symposium, you have clinicians, basic biologists, people who work on MRI, people who work on genomics, people who work on metabolomics, all kinds of people come. So let's say if Taru and Prasad are two people who are coming, if they want to collaborate, they'll collaborate. If they need a particular mouse model, we'll provide a mouse model. If they need samples, we will connect them with clinicians to build cohorts. If they want technology, we'll provide technology. Who goes first author, last author is their problem. They can fight with each other. Me and Mahendra don't want a publication. If we are only intellectually involved, we want a publication. Otherwise, you only have to thank Arumda. We truly want to make sure that we don't drive the science that happens in Arumda. People drive science. We only provide support. Of course, in projects, some of these projects we are deeply involved, we will publish. We have enough publications for our life. We will continue to publish even if Arumda didn't exist. So that means we are not looking at this as a publication measure. The second thing is, for the first time in my view, particularly in India, definitely it happens probably in the West, Arumda became a key partner in one of the largest diabetic conferences in the world, which was a joint effort between Albert Einstein College of Medicine in New York and CMC Vellore. This had an important outcome, which is going to define how we think about diabetes. Now, lean diabetes is very common in India and Africa and entire South Asia, right? Lean diabetes is not type 2 diabetes because there is no insulin resistance. It's not type 1 diabetes because you don't have beta mast cell destroyed. You have low insulin, low muscle mass. We don't know exactly why this happens, how do you treat it, what are the markers. So International Diabetic Federation and American Diabetes Association, both presidents were present in this particular meeting. And together, Arundas, MC Vellore, and Albert Einstein College of Medicine, we've come up with a declaration to identify malnutrition-induced diabetes as type 5 diabetes. That means there will be new funding, new opportunities to make sure that we understand this diabetes, because in lower income countries, this is the predominant diabetes. Of course, ultimately, everybody becomes type 2 because you develop insulin resistance over a period of time. But this is an important thing. So this is really one of the major milestones that we have achieved. The other thing is, now remember I said stable isotopes. Stable isotopes is not about only body composition. For example, if somebody becomes obese, do you know, is it the fat from diet or the fat that is produced in the body? You know, there's something called de novo lipogenesis. Fat is produced by the liver and in the adipose tissue. How do I know how much of this fat was because of the glucose that got channelized into fat, or your fatty acids coming from your diet? I can drink D2O, and deuterium gets incorporated into the fat that gets synthesized. If I measure palmitate, all labeled palmitate will be because the fat that got synthesized from my body. Unlabeled palmitate will be my dietary fat. I can actually know whether you're becoming fat because you are producing more fat or you're consuming more fat. Okay, but I can do this many ways, including by measuring iron uptake, protein turnover. I can give labeled alanine and look at this. And these are non-radioactive C13 labels, very safe to do this. Now, if I can do this, of course, Arunda will flourish. We will do very well in India. Maybe my collaborators or our collaborators will do well. People in the Arunda network will do well, but that's not our motivation. Our motivation is to really empower. Remember, we are empowering Anganwadi workers. We also want to empower the researchers. So it was again two-way, like it happened with ICMR. International Atomic Energy Agency came to us, or we went to them. It was more like a mutual match finding. It's like a Tinder, whatever it is. Do you guys have Tinder? I don't. Okay, no need probably. IAEA came to us saying that, look, whatever we have done so far is giving you scattered data around the world, which means we are not able to make a policy impact because it is scattered with small pockets of information. So what is Arunda doing? We have brought in so many people together. We are harmonizing studies. That means, otherwise what used to happen is somebody used to do some study in Delhi, somebody used to do some study in Chennai, somebody used to do some study in Mumbai. All of these studies would be differently designed. Therefore, I couldn't compare the results. If I can compare the result, what is the parameter here is an exclusion criteria here, right? So that means, how do I come up with a policy? I can't come up with a policy. So we are trying to harmonize. In fact, on May 2nd, we are having a meeting of all people all the people in the country who have done what are called birth cohorts, which is looking at small for gestation age, preterm birth, blah blah blah, to say, can we now start building a longitudinal cohort? That's something we are doing. So we said, if we can do all of this, we also want to empower our partners to do this kind of techniques. So we now do annual workshop on stable isotopes, and this is available not just to people in India, but entire South Asia and Africa and probably global South. In fact, in this particular workshop that we organized this year, we had people from Thailand and other countries, right? That means we want to train these people to do these things. It's not about whether you publish a paper or not. Maybe they'll publish a paper. Maybe they'll forget Arunda after 5 years. It doesn't matter. But they are doing something relevant for their population and they're helping their population. So that's something that we are doing here. And all of this wouldn't have been possible without an amazing bunch of students. I don't know if your peers tell you, we love students. We do scold them, but we also love them. All students work hard. My students probably work harder. I'll tell you why. When I joined TIFR, we didn't have an animal house. The only animal house that was immediately available was in Actrek, which is 45 kilometers from Bombay. We were in South Bombay. We used to go to Actrek, which is in Kargar. Then at some point in time, the Actrek director said, we need animal house for ourselves, shut down. And then we said, what do? We went to ISA Pune. So most of the publications that I showed you on mice, which came between 2017 and 2022, all experiments happened in ISA Pune. We used to go every day, me and my students, carry dry ice, and people in ISA Pune used to call me Coolie PI because I used to carry dry ice and tips and water and everything on my head. My students have done this. I don't think many students will be up to. It's not because I insisted on that. Even now, I'm hardly there in my lab. My lab works. They are motivated, self-driven. I'm really glad for them. So I used to become more emotional when I used to talk. I've become less emotional these days. It's thanks to them that I'm even standing here, and all credit goes to them. The fact that we could even dream about Arunda is because they are doing their science in their lab. Of course, a lot of funding support, a lot of facility help, great collaborators that I have, and thank you everyone for your attention.
Thanks, Ullas, for the wonderful talk and a lot of insights with your research as well as with Armada. So people might, maybe they're filled with a lot of questions,
Yes.
maybe early and later, whichever it may be, any part you can choose as you wish.
I have two questions.
Yes.
So the first question is on the fly experiment that you said you are sending them to the space. Are you also looking at the transient effect on the next generation, meaning epigenetic effect on the subsequent generations as well?
We wanted to. Actually, we asked for larger, all payloads are extremely tightly controlled because everything that you add adds to the weight. So it's a real estate problem. Actually, ISRO says real estate. They said you will get a real estate out of this. So utna kitna experiment kar sakte ho karo. So actually, we wanted to send more flies to look at next generation, but we couldn't manage. We had,
Once you get back the flies, are you getting back the flies?
we are getting back the flies, but
Can't
they'll
you
be
breed them
already
here and
they'll
then-
already be old.
Okay.
And usually flies, they like to reproduce as soon as they close, and then usually you do within first 5 days. After that, generally there is babies are sick, or let's say larvae are sick. So yeah, ideally we could have done it, but no, maybe that will happen in the space station.
The second question is on the larger population study that you were talking about, 8,000 children, something you were saying. You're going to get data from, every parameter that you're looking at.
Yes.
I'm just curious to understand, pardon my ignorance, I'm
Sure.
just curious to understand how do you, think about the ethics? For example, you are studying a particular population where you think they're not eating right. Still, would you avoid advising them to eat well or give them a proper plan because it's against your study, or how do you manage that?
Good question. So this is not for the reference standard growth. It's not a problem because we are not selecting a malnourished population. In fact, the WHO growth standard is based on only urban populations. And ICMR is also separately doing, in fact, they were supposed to identify 7 other populations who will work with us, but they only selected first 1,000 days. They said it has to be, middle to upper middle class households living in cities, blah, blah, blah. My worry is that they might be actually not the correct reference standard. They might be actually their childhood obesity, right? So therefore, we said we'll work with both rural and urban population. In fact, ours is the only study which compares in parallel urban and rural at this level, right? Therefore, we are not looking at anybody who's malnourished. We are keeping an exclusion criteria that somebody should not be sick because of genetic diseases. Somebody should not be sick because of a maternal or a paternal disease. These we are considering children who are supposedly healthy. We're capturing all the data, and now the metadata will be used to ask how much of nutrition, how much of physical activity, how much of smoking at household, or blah, 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 blah. So we're going to do that. But in the micronutrient supplementation study, that's what we are doing for adolescent girls. Now, in adolescent girls, the problem was that, for example, Gates Foundation said that we have this what is called a Unimap concoction, or you have what is called MMS, multiple macronutrients. So usually these are given as supplements. They said, there is a risk of over supplementation. Iron toxicity is as bad or worse than iron deficiency, right? So we said we don't want to blindly give adolescent girls all macronutrients. So first we're going to identify phenogroups within this, and we're going to supplement them. Yeah, ethically it becomes difficult. Whom do you supplement? Whom do you not supplement? Usually, when such happens, you give one RDA supplementation because somebody. But we can now play around with different dosing to ask whether they are getting better or not. Yeah, this becomes very tricky. Very good question. It is what it is. We have to deal with all these problems in real life world. Yes. Yes. Thank you.
It's a fantastic talk. When you speak about neurological memory, there's a concept called engrams. So they are stored physically inside the brain in certain parts, and over the time they might get lost, but it can be recalled or understood later by some optogenetics or fMRI based things. So the memory of biological mechanisms, can it be recalled? If you're saying that the memory is getting lost and that is what is causing diseases. So is there any mechanism or a physical storage of this memory that happens?
Yeah, so absolutely yes. So this physical storage of memory is not only the neurons. In fact, I keep teasing my neurobiologists that just because you work with neurons doesn't make you a brilliant, more brilliant than somebody who's studying leaf, right? Biology is biology. Just because, but sometimes neurobiology, oh, neurobiology, acha, tum plant pe kaam kar rahe ho, right? We do think about that. I don't know if you think about it. Anyway, let's get back to the question. Physical memory can be stored. For example, chromatin conformation can be my physical memory. Suppose if I drive a particular enhancer promoter interactions more because I'm on a particular lifestyle, that might get more solidified. I might form a pad there. That pad might become entrenched and fixed over a period of time, lose its plasticity, and become a memory. But I can invoke many such mechanisms in biological systems. Okay, some less stable, some more stable. But the most stable ones would be my chromatin. Right? I hope I answered that. Membrane architecture, cell-cell interactions, tight junctions, all of those could become part of memory. Yes.
Hi, Ullas.
Yes.
Yeah, very nice and expansive talk. My question is on your starvation and refeeding. So you have looked at refeeding, but what happens in starvation? Is it exactly the reverse or something else that happens? And starvation, I believe, is slower process than refeeding.
Yeah, so I didn't get time to go through this. In fact, that's exactly why we said starvation is a programmed event, right? It happens at hours, minutes, hours, days, weeks, probably in some animals, 2 months. In fact, a paper that we are writing now actually shows what dictates first gene should be gluconeogenetic. First gene should be glycogenolytic genes. Second gene should be gluconeogenic genes. Third gene should be lipolytic genes. Who organizes these cascades of transcription? We identified intrinsically disordered regions in proteins that dictates these interactions to give you a programmed skipping or programmed handover of transcription factors on the same genes. Okay. So therefore, we work a lot on starvation. In fact, I didn't have time to look at it. We look at exactly how programming is happening during starvation. And of course, what happens in refeeding.
Yeah, the other question is this starvation refeeding that you're doing, have you looked at different strains of mice?
Yeah, we have. There are subtle differences, but largely starvation is starvation. Now, whether you starve a cattle or a human, we'll all induce lipolysis. So therefore, to that extent, or a fly, okay, you have these evolutionarily absolutely mandatory responses that you have to mount being any organism, which are these gold standards, right? The subtle variations can vary. The subtle variations are important. I'm not saying no. In fact, we use what are called C57 black 6 N mice. Most people use J mice. J has an NNT1 mutation, right? So most of the biology literature is littered with J mice. We only use N mice precisely for this reason because we don't want to have this. Yeah, but we have looked at multiple strains. This starvation response and refeeding response, same microRNA may not come up. For example, if I say MIR let7I, it could be let7A. But the fact that MIRs oscillate is concerned.
So,
Yes.
hello.
Yeah.
My question was similar actually. So I wanted to know if there were any studies done on hibernating animals because they have a drastic change in the fat composition and everything. So
yeah, people have done a lot of studies on hibernation. Mole rats are one thing. A lot of studies. But hibernation is not the same as feed and starve response. Right? You're feeding and starve. Even hibernating animals, when they are not hibernating, go through feed and fast cycles. Right? So hibernation is something which is more long-term programmed. We don't even understand that very well. Right? Again, it involves epigenetics. It also involves mitochondria. So there is literature where people have done global transcriptomics, global metabolomics. But genetic intervention is difficult. You can't also intervene them because then it's not easy to uncover. Yeah, but hibernation goes through similar pathways, but the response is very different. You still need the same PPR alpha. You still need the same FOXO. You still need the same TFAM. You still need the same Paul gamma to induce all of this. That's what I said. The genetic components are the same. How they come in space and interact with each other is different.
And one more question. So, to ma'am's question, you had said that the genetic history and the environmental cues that when they're in conflict, it leads to metabolic diseases. So, I read about some some articles. Basically, I'm not sure about the validity of the science, but they claim that due to the repeated periods of famines and other things in human history itself. So some populations are more attuned towards like accumulating resources during the periods of plentiful. We could say.
There's a very interesting evolutionary concept to this. This is called thrifty gene hypothesis.
Yeah.
Right. And it seems to be true. Right. Why do all of us humans? I don't know what Taru is going to feed us after this talk. I'm hoping, not hoping there is a sweet there. Even though we know that sweet is bad, why do we consume it? Because evolutionarily, it's encoded that we were always in a resource-constrained environment. No organism has unlimited access to food in the wild. So, evolutionarily, the moment you see food, you're supposed to consume it so that you store it when you need it. Okay, that's the whole principle of the general feed and fast meals. Therefore, feeding and fasting is very important. The durations of feeding and fasting is also dependent on your overall physiology, but also in humans, it becomes very cultural, right? So therefore, it becomes very complicated now to ask these questions, but we can ask biologically, right? In fact, in humans, we can do these perturbations about feed and fast cycles. That's why intermittent fasting is so popular these days, right? Because again, it triggers the same feast-famine cycle concept. Thrifty gene hypothesis seems to be true. I hope I've answered the question. Yes.
So that is why there's a difference when you exercise and when you do the dieting. So the dieting is a wrong concept because exactly what he said. So if your body doesn't get and suddenly it gets, so it just says give. So then instead good bad happens, and that's what he was showing the experiments of mouse on the treadmill. So that is towards the muscle mass and whatever you said, that sarcoplasm and stuff. It's this way. I think
Yes,
I have a
absolutely.
point.
So we are doing very interesting study now. Many muscle builders or many people who exercise drink a lot of whey protein, right? They do a lot of glutamine supplementation. There's glucose Actually, that might be bad because if you consume a lot of glutamine, you know what is called aneuploretic flux. In the TCA cycle, your glutamine feeds your TCA cycle. That means you go to deamination reaction. You build a lot of nitrogen. Exactly. And liver is also burdened. Now, you think you're building muscle mass, but in the long term, you might be actually doing a lot more damage to your system, which you don't know. So you're actually doing glutamine supplementation and protein supplementation to mice when they exercise in the morning or evening. We're also making them exercise at the wrong time of the day, doing reverse phase exercise. That means inverting the circadian cycles.
So I have a very simple question with the Armuda.
Yeah.
Basically, you're talking about stable isotope labeling followed by tracing. It's been 1.5 years. We are unable to get the gamma ATP in Hyderabad. How it is feasible?
Gamma ATP is not my world.
No, I'm telling isotope labeling. You
No,
take it anything, like it's been almost more than 1.5 years.
That's for all of us, right?
Yes, exactly.
This is now because of an AERB and a BRIC problem, that is another BRIT problem, right?
Yeah,
BRIT problem.
Yeah,
BRIT
you are
problem.
right.
So until 2 years ago, I used to be in all or some of the important committees in DAE, where I had possibly the reach of people whom I could talk to. Unfortunately, many of them have retired now. Okay, but I'm given to understand it's almost solved.
Yeah, we heard that it's been 6 months. They were telling same thing.
It's almost solved. It was basically what happened is that even to make this gamma ATP, you need to import. That import ran into a problem. Okay, and it was mostly a financial administrative problem that created this. It's not DE wanted to stop it. They couldn't source the raw material, and therefore they had a problem. But now I think it's almost resolved.
If no more questions, thank you very much for the wonderful talk.
Thank you very much.
Give him a big hand, guys. And now I request Director Ma'am to facilitate our today's speaker, distinguished guest. No, just to facilitate. Yeah. Come down. Yeah. We can have a picture also later together. Now I request Dr. Patil to give vote of thanks. Yeah. After that, we'll have a picture here and don't run away. Okay. After that.
Yes, wait.
Yes, yes. Also. It is there. We kept as he said that it's not a cola, but at least fruit juice is there. It's a
So-
sweetener.
Good evening, everyone. As we come towards the end of this program, I have the pleasure of giving the vote of thanks. On behalf of the Scientific Events Organizing Committee, I take this opportunity to extend our heartfelt gratitude to all those who made today's distinguished lecture a great success. First, we sincerely thank our director, ma'am, for her support for having this lecture, wonderful lecture here. Thank you, ma'am. We thank Professor Ullas Kosrul Sir, Director, CDFD Hyderabad, for delivering such an insightful and thought-provoking talk. Thank you so much, sir. It was indeed very nice. Your work and perspective have truly enriched our understanding and have inspired new thoughts for research and application. A special thank to all colleagues as well as to the members of different domains of NIAB, stores purchase, ICT, administration, finance for their support. A special thanks to all our students and faculty members for their enthusiastic participation and engaging questions, which has added great value to the session. We also extend our appreciation to different sections for their seamless coordination and support behind the scene. And last but not the least, the members of the committee for having this seamless program throughout. Thank you all. I now request everybody to gather.